Hello, ABS. Thank you so much for coming by to check out my talk. I am thrilled to be a part of the Elise Symposium this year and to tell you about my research on the cognition of urban wildlife. Urbanization represents a dramatic environmental transformation that has been linked to biodiversity loss. This is largely because urbanization is accompanied by an increase in humans and human entities, as well as a decrease in natural resources. Nevertheless, we see that many species persist in urban environments, and although there are several factors that will contribute to a species' ability to tolerate urbanization, one outstanding factor is the use of cognition, specifically the use of cognitive abilities that allow animals to overcome novel or complex challenges in their environment. So the notion of the importance of cognition in overcoming environmental challenges stems from the cognitive buffer hypothesis, which proposes that large brains have evolved to facilitate domain general cognitive abilities like learning and problem solving that allow animals to produce new or modified behaviors that will increase their survival and by extension their fitness. And it's thought that this entire process is fueled by new, unusual, or complex environmental conditions. So a popular question right now in the field of cognitive ecology is understanding whether urbanization is acting as a selective force on the cognition of wildlife. So the cognitive buffer hypothesis is largely supported by correlational evidence that demonstrates a relationship between brain size and various metrics of ecological success. So for example, across mammals, birds, and amphibians, a relationship has been found between a species' relative brain size and its ability to invade a novel environment. Within the context of urbanization, there is some experimental evidence that suggests that urban individuals are better learners and problem solvers compared to their more rural counterparts. However, results from studies such as this are pretty mixed, and they're mostly limited to birds. So right now we're at this point where if we want to better understand if and how urbanization is acting as a selective force on the cognition of wildlife, we need to not only continue developing reliable ways to study cognition in the field, but also extend our testing to non-traditional but highly adaptive species. Raccoons stand out as one of these urban species that likely relies on cognition for their tolerance and successful exploitation of urban areas. They are a widespread species, invasive in many parts of the world, and they're often considered to be a nuisance. They are habitat and dietary generalists, skilled in extractive foraging, and they have some pretty impressive brains. So for example, we recently learned that the number of neurons in the raccoon brain is more similar to that of primates rather than other carnivore species. So based on what we know about raccoons and their natural history, as well as anecdotal and captive observations of raccoons, we can expect raccoons to be rapid learners, who are behaviorally plastic, and that they are innovative problem solvers. And it's likely that these cognitive traits aid in their heightened ability to exploit urban environments. In 2015, I began working with the University of Wyoming Raccoon Project to study the cognition of raccoons living in the city of Laramie, Wyoming. Every year, we humanely trap raccoons in order to collect information on their age, their sex, their docility towards human handlers, and most importantly, to mark them with pit tags, which allow me to deliver tests of cognition in the field using radiofrequency identification techniques. And to date, we have trapped and identified over 200 raccoons. Today, I'm going to tell you about two of my field experiments that evaluated the cognitive abilities predicted to be essential for urban tolerance. So the first experiment investigated innovation and learning using puzzle boxes. An innovation is the creation of a new behavior or use of an existing behavior in a new way. And because it's really difficult to capture the onset of innovations in the wild, researchers often rely on puzzle boxes. So a puzzle box is an apparatus an animal must figure out how to open in order to retrieve a food reward. And by watching the manner in which the animal engages with the puzzle box, as well as how long it takes them to open it across trials, we can quantify learning and innovative problem solving. So here's a learning curve taken from my advisor's previous work with hyena. And as you can see, the amount of work time to open the puzzle box decreases across trials and produces this nice negative slope to the learning curve, which demonstrates learning and innovative problem solving. For my research with raccoons, I designed a puzzle box with many doors on it, so I could quantify the amount of work time from the opening of one door to the next, which would allow me to calculate a learning curve based on one visit to the box if necessary. I placed the puzzle box inside of an RFID antenna, so if a pit-tagged individual approached the puzzle box, its identity would be recorded. I also placed 4 night vision cameras around the outside of the puzzle box so that I could record all trials, and then later go back and match up an individual's identity to its video footage for scoring. I also used a second puzzle box to specifically assess repeated innovation, which can be a metric of flexibility and problem solving. And this design is different in that the puzzle box can be opened in a number of different ways. And once the animal figures out how to open one of the solution types, that solution is locked, which then forces the animal to innovate again in order to get to the food inside the box. Now, the multi-axis puzzle box design you see here was used by my colleagues in a recent study with captive raccoons. And they found that if a raccoon was ever going to solve the puzzle box, they always found more than one solution type, which demonstrates repeated innovation. To assess repeated innovation, I modified my single solution puzzle box so that it contained 4 different solution types. And the idea here is that once the raccoon has opened all of the doors that it knows, it must change its strategy in order to access food behind the remaining doors. So here is a single solution puzzle box trial. We're looking at an adult male raccoon named Grand Teton, and as you can see, he just opened one of the doors. And if we fast forward a bit, we can see that Grand Teton is joined by a group of juvenile raccoons, one of whom knows how to open the door by sliding the bolt latch to the right, which will pop the door open. Now this is footage from a multi-solution puzzle box trial, and although it's been sped up in the interest of time, you can see that this individual is moving through the various solution types and is able to get to the reward behind each door. With the help of undergrad research assistants, Carissa, Cooley, and Emily Davis, we tested 35 wild raccoons, 8 of whom learned how to solve the box. In this figure, I have the first 60 door attempts on the x-axis and the mean work time to open on the y. And as you can see, the amount of work time significantly decreases across doors, indicating that raccoons learned to solve this novel task over time, and they became especially proficient at solving after opening 25 doors. We also found that exploratory diversity, which is the number of unique behaviors directed towards the puzzle box, was the best predictor of problem-solving success, and this is similar to many other studies of innovative problem-solving. We tested 31 raccoons with the multi-solution puzzle box, 7 of whom learned how to solve. And our results are very similar to the captive study I mentioned earlier, in that if a raccoon was ever going to solve the box, it always found more than one solution type, which demonstrates repeated innovation. But I really wanted to dig into the solver's results a bit more, and initially I felt pretty limited because I had to modify the typical multi-axis puzzle box approach to meet the needs of my wild system, which meant that I couldn't really apply the typical analyses to my results. So I forged a new way to make inter and intra-individual comparisons of flexibility using a sequence analysis approach. So for every trial, we recorded the sequence of solution types opened, which you can see visualized in this bar here. So every color in this bar corresponds to a different solution type. And by working with the data in this manner, I found that we can do a couple of pretty cool things. So first, we can get at flexibility by quantifying the number of transitions between solution types and combining that with the frequency of solution types found to give us what's called a complexity index, which ranges from 0 to 1. So 0 would indicate no complexity and that the animal only found one solution type, whereas a 1 would indicate the maximum complexity possible and would indicate that the animal found all of the solution types in equal numbers and that they were switching between different solution types every time they opened a door. So after building sequences for all raccoons for all trials, I found that raccoons have pretty high complexity to their sequences. However, this was dependent on their prior solving experience. And you can see what I mean here. So in this figure, I've plotted the complexity scores for knowledgeable raccoons who solved the single solution box and naive raccoons who did not solve prior to the multi-solution trials. On the x-axis, I have trial number, and on the y, I have complexity score, which again is my proxy for flexibility. And we can see that the knowledgeable raccoons in red had high complexity from the start, indicating that they were able to generalize solving to the new solution types. However, our naive raccoons in blue started with lower complexity scores, but increased their complexity after gaining solving experience across trials. So I think these results illustrate how learning begets learning, and it can be an important part of the development of flexible behaviors. Now, the second thing that I've done with these data is ask how similar performances within and between individuals using optimal matching, which is basically a measure of dissimilarity between two sequences. So I've made pairwise comparisons of all of the sequences within an individual's own trials and also between the trials of different individuals. Starting with the within individual comparisons, there appears to be some dissimilarity across trials, indicating that individuals were not opening the same order of solutions every time they were at the box. But if we look at the between individual comparisons, we can now see that there is greater dissimilarity between individuals compared to within individuals. Now, these data I'm showing you here are specific to trials where individuals were tested alone. But we had a lot of trials where multiple solvers were present, which created a more competitive testing environment. And if we look at these times of competition, we can see that there's certainly variation. However, the same pattern emerges where we see that there's greater dissimilarity between individuals rather than within individuals. In fact, raccoon performance was more self-similar when constrained by competition compared to times when they were alone and their performance could be unconstrained. So to me, this shows that raccoons demonstrate both preference and flexibility in their problem solving, and this provides new insights into the foraging tactics that animals use when in groups in the wild. So my observations of flexibility in raccoon problem solving led to my next experiment where I assessed the cognitive underpinnings of behavioral plasticity. So behavioral plasticity is an animal's ability to change its behavior in response to changing environmental stimuli. And the most traditional way of assessing the cognition behind behavioral plasticity is by using reversal learning tasks. And this is where an animal is exposed to a positive reinforced stimulus and a negative unreinforced stimulus. And once the animal demonstrates that it can discriminate between the stimuli, the significance of the stimuli are reversed. So the previous negative stimulus is now the correct response. And behavioral plasticity can be assessed by totaling the number of errors that the animal makes before it starts consistently selecting that new correct response. And it's thought that animals that are highly plastic or flexible in their behavior are going to make fewer errors during this transition, whereas animals that are more rigid are going to make more errors. Recently, folks within the bird world have been able to bring tests of reversal learning into a field setting by using automated feeding devices equipped with RFID technology. So I adapted this technique for raccoons by working with Eli Bridge and Joost Huizinga to create a testing device that delivered automated reversal learning trials. So when a pit tag raccoon approached the device would scan the ID of the raccoon, deliver tests specific to that raccoon, and would keep a record of that raccoon's performance across time. So here are a series of video clips taken from the field to show you what our testing protocol looks like. So every night when we put out the boxes, I program them so that they will first dispense a food reward anytime a pit tag is detected. And I do this to habituate the animals to the testing device and to encourage their participation with it. But after they've depleted the freebie rewards, they have to start pushing on the buttons in order to trigger the release of a food reward. And so you can see them learning to make that association here. Here we have a raccoon that makes a correct response, so it gets a food reward. Here a raccoon makes an incorrect response, so it is now in a brief timeout period, and then it's able to alter its behavior and make a correct choice. Now we saw a fair amount of competition for access to the box, which was at time super adorable, but it unfortunately created some disturbance in our testing. Jumping into the results, we found variation in the way raccoons responded to the testing device. Some raccoons were very spooked by the device and they never engaged with it, whereas the majority of raccoons either came night after night and collected their freebie rewards, or they actually learned how the device worked and they participated in trials. We found that juveniles were more likely to habituate to the testing device. However, they typically performed worse in reversal learning compared to adults. We also found that raccoons that were more docile to human handlers, which was measured as the production of aggressive vocalizations made during testing and processing, were more likely to learn how to operate the testing device. We use linear models to assess performance at both a group and an individual level. And in this figure, I have reversal number on the x-axis and the number of errors being made on the y. And it does appear that at a group level, raccoons are reducing the number of errors being made, which is what we would expect to see. But if we look at our individual learning curves, we can see that not all raccoons improved across time. And if we compare these results to data that we collected in captivity as a part of another study, we can see that the captive raccoons outperformed the wild raccoons as evidenced by a greater negative slope to their learning curves. And so I think this difference in performance that we see is likely due for the need of wild raccoons to remain vigilant and that they had more interruptions in their testing in comparison to the captive raccoons. So in sum, we have successfully adapted cognitive testing protocols and developed new analyses to assess the cognition of raccoons in the wild. And this type of research is at the forefront of studies in cognitive ecology. We found that raccoons demonstrated rapid learning and repeated innovation in our puzzle box experiments, and our results are similar to that of previous captive research. We also found rapid learning and behavioral plasticity and reversal learning. However, wild raccoon performance was susceptible to social and environmental disturbance, so these types of tests are going to require further refinement. And finally, we found individual variation in participation and performance that was due to factors including age, personality, and the social environment. So this research does offer some support for the cognitive buffer hypothesis in that raccoons demonstrated the cognitive abilities that are predicted to aid in urban tolerance. However, there are some limitations to this research as I was working with a single population. So an obvious next step would be to test additional populations in higher numbers to gauge how generalizable the results are. And of course, linking the results of cognitive experiments such as this to fitness benefits will generate greater evidence for selection on cognition in urban areas. And looking forward more broadly, I'm really inspired by this current shift that we're seeing in urban and cognitive ecologists, and that we're moving away from thinking about urbanization as a single evolutionary pressure, but rather a more complex and multidimensional force. And I think this is important because the challenges imposed by an urban environment and the subsequent cognitive strategies that animals use are going to vary by species and be shaped by several features of a built environment, including human attitudes towards wildlife. So at this point, I think we need to really embrace urban environments as complex social ecological systems and work to delineate the specific challenges that animals face in a given urban environment if we want to better understand the role of cognition in urban adaptation. And with that, I would like to thank my co-authors and my collaborators for contributing to this research. Again, I'm thrilled to be a part of the elite competition this year, so I'd like to thank ABS and all of you for listening, and I would love to take questions in the Q&A. Thank you.